Okay, for this immobilization, easy if I just talk about it is how you attach. But most of the time we use immobilization, means that we want the bioreceptors or chemical receptors to be on the surface of the sensor, and we want them to be stable, yeah, on the surface of the sensor. And this is a very crucial part, yeah, in sensor development. Very, very crucial part, because if we do not do it correctly, it will affect the performance of the sensor. Okay, it will affect the performance. So never underestimate the presence of the surface. It's about surface chemistry. Okay? Development of biosensors must take into account the effect of the local environment. Means that if you are to test the sensor in, for example, in blood, in river water, or in food matrix, we have to remember the environment is not that kind. The environment is very complex. Okay, so we must make sure that whatever bioreceptor or chemical receptor that we are using must able to withstand all this interference from the surrounding. Okay, so it affects the local environment, the binding capacity of the immobilized selective molecular. Okay, so we have to we have to make sure that whatever that we put on top of the sensor surface will be able to be stabilized, even though they have harsh environments. It will not break down the attachment on the surface of the sensor. Farah, you have issues with your internet? Okay. So in order to understand this surface chemistry, okay, we are talking about surface chemistry. We need to know, one is of course the nature of the solid substrate. Means that what kind of surface are you? What kind of material are you using? Okay, we have carbon, we have gold, we have silicon. Those are quite common things that we use for sensor surface. In my case, there's others as well. Right. And then that leads to what method that we are going to use to attach, to immobilize the bioreceptor. Okay. And we also need to think solution conditions. If we are to talk about medical related diagnostic, especially when we are doing DNA or antibody, we need to think about the temperature suitable for the antibody, for the DNA. If it is enzyme, what is the suitable temperature? And also what is the suitable pH? Yeah, that's why the usage of buffer and the usage of temperature, the usage of the study, yeah, the study of buffer and the study of temperature is very important in making sure that whatever that we attach on the surface of sensor will be very stable, okay, and optimize the interaction of the receptor with the analytes. So that's our target. So these two actually very important parameters. Yeah. Probe effects, because I'll be using DNA, a lot of DNA for the example of this immobilization. So as we have studied, as I have discussed about DNA probe before, for DNA based sensor. So DNA probe can be it can be 30, what we call as 30 mm long, okay, or it can be 25. What does it mean? It has 30 amino acids, or it can be 25 amino acids, or it can be more than this. It can be 40 or it can be 70. Okay, the sequence of amino acids in that DNA probe. So there are some advantage and disadvantage using short or long DNA probe sequence. Yeah? Depends on the, if it is too long, it might get tangled. If it is too short, it might be not that selective. So it has to be balanced, right? So now, one of the consideration that we have to make in choosing what strategy to attach the receptors, okay, we have to reduce the restriction. What does it mean? We do not want the bioreceptors or the chemical receptors to be too crowded. Okay, we put too much on the surface of the sensor, and they sort of have this uh, restriction. Okay, it's difficult for the receptors to have space for interaction. Okay, so this has to be studied. How much is how much? How much is enough? How much will give you the best signal? So this has to be studied at the early stage of sensor development. Okay. Stable immobilization, of course, we want it stable. We do not want the receptors to leach out or to be removed from the sensor surface. Yeah, we do not. We want stable chemistry. And accessibility must be very important. That's why we try to reduce the restriction. Okay, I hope that you remember that for DNA-based biosensor, the target DNA has to come close to the DNA probe to be able to hybridize with the DNA probe. And for antibody, the antigen has to come close to the antibody to be able to interact with the antibody. Okay, same goes with aptamer. The analyte has to come close to the aptamer in order to interact with the aptamer. So if we have a very restrict situation and the analytes of interest cannot come close to the receptors, then it's difficult to get signal. Okay. And there is another issue called non-specific binding. Okay, so as we know, there's a lot of other things exist in the sample. Okay, like if we have blood, if we have sputum, if we have urine, we can imagine there are other proteins, there are other compounds. Okay, that is also will be interacting. So how do we limit the non-specific? Means that the one that we do not want to attach to the receptors. We only want the specific one. Okay. This also has to be resolved. Okay. And while preparing the attachment of the receptors, we can at least prepare to strategize in reducing the non-specific binding. Okay. Yeah, this is, I think, the same. Irreversible means that it is stable, non-specific binding. Okay, right. So the immobilization or the attachment, okay, all right. Uh, there are many ways to do it. There are a lot of strategies, a lot of chemistries, a lot of chemistry that can be used to attach. Okay, but for general, okay, this is what we are going to learn. A different strategy to attach the receptors on top of the sensor surface. Yeah? Transducer surface means is the sensor surface. Yeah, we have what we call a simple adsorption. We can use polymer, and there is what we call as biotin streptavidin, biotin streptavidin chemistry. Okay, nanoparticle, covalent attachment, and this is a self-assembled monolayer called SAM. Okay, so these are among the general strategies that we can use to attach the receptors, okay, on top of the sensor surface, whether it's DNA, aptamer, or antibody. Okay, so if you have like different kind of receptors, for example, enzyme, then probably you have different strategy. Okay, I will talk about possibility of different protein like enzyme or chemical marker, how you can attach them to the sensor surface. Yeah. So attaching a sensor to sensor surface is not really a complicated process. We can use a simple lab-based process, or we can use an instrument. Okay, so if you are interested, I think I have given some option, which is face-to-face in the calendar. So face-to-face means if you're interested to look at the process, you can make an appointment, and then I will show you some examples of how we attach the receptors on top of the surface, sensor surface. Okay, so the first strategy is simple adsorption, and usually this is using carbon, carbon surface. Okay, it's not necessarily electrodes, but it can be optical, it can be other types of transducers. But since carbon is very absorptive. Okay, its capability in absorption is very high. So we can just incubate the bioreceptors or the chemical receptors in a solution, and then we put the carbon, whatever, whether it's electrodes or optical, and they can naturally adsorb, naturally adsorb on carbon, because carbon can absorb a lot of things, So what you can imagine is, if we have a carbon surface, okay, so if we just put this carbon electrode, for example, incubate in the beaker, it's a simple process. So let's say this is carbon electrode, and then you put the DNA probe. Okay. So as we know, DNA has these negative charges, phosphate backbone, and they also have amino acids. Okay, all these charges will interact with the carbon surface. It will just not interact in terms of chemistry, but physically adsorbed on the surface of carbon. If we are talking about chemical receptors. Yeah. And the interaction of the chemical reagent with the analyte is not really orientation sensitive, then it's okay. I mean, the analyte can come close to the whatever receptors that you have put on top of the carbon, and they can interact. But if we have orientation sensitive, like DNA, okay, it has to stand up, for example, or like antibody, it has to stand up, or like aptamer, it requires a specific orientation, yeah, specific orientation, then this method is not suitable. Okay. So let me give an example. If we are working with enzyme, okay, let me get on. Yeah. Okay, let's say I'm talking about using enzyme. Yeah. So enzyme is a protein, and it's a bulky protein. And let's say I just want to put it on a carbon surface. So you incubate the enzyme, okay, together with carbon electrode, let's say. Okay. So it will be, this is the carbon electrode, and you have the enzyme, and you expect the enzyme will adsorb, yeah, on the surface of the carbon electrode. So since enzyme, it does not have a very specifically requires certain orientation, then whatever substrate, let's say glucose, and this enzyme is glucose oxidase, then the glucose will just interact with the glucose oxidase, and there is certain peroxide generated from the reaction. Okay, so there is no need for us to use complex chemistry, because it's quite easy as long as the enzyme is stable on top of the carbon electrode. Okay, so when I say orientation, okay, okay, so this is example of DNA, right? Okay, so if we use like simple adsorption, okay, simple adsorption, okay, and we know the phosphate backbone is negative in charge, so they will tend to lie down. The DNA will tend to lie down. So the problem is when the target DNA trying to interact with the DNA probe, then there is a difficulty because the orientation of the DNA is not that optimum for hybridization purposes. So imagine if we can have the DNA to be standing up, then it's quite easy for the target DNA to approach the DNA probe, okay, for the hybridization process. Okay, we have discussed about hybridization process. So this is what I meant by orientation sensitive. You have to think about whether you need the orientation to be correct in order to have a good interaction, or you can just have simple adsorption to happen. Okay, so that's the disadvantage of using simple adsorption. Yeah, simple adsorption. Okay, so if you don't need orientation, then you can use simple adsorption. So this is the issues with adsorption. If we have hybridization efficiencies are low, then the detection limits is not good. Okay, so greater chance of errors. So for DNA, for example, or antibody, this is not preferred. Yeah, technique. So the second one is polymers. Yeah, here can we use polymer to hold or to attach the bioreceptors or chemical receptors. Okay. Again, in this case, this is the common my battery is out. Okay. Right. Okay. It's okay. I think I just charged it just now. Okay. So polymers. Charge. So if you look at the picture, the diagram here, you can see that the DNA probe is immobilized in the middle of polymer porosity. Okay, so one of the example using some polymers can be used is polyaniline, polypyrrole. So if you look at these polymers, they are positive in charge in terms of aniline, in terms of pyrrole functional group, they have this positive charge. So it's good because they can interact with the DNA negative charge on the phosphate backbone. But again, if you try to imagine first, why my yeah, okay. You can see that there is difficulty as well if the DNA target trying to approach here. Then the DNA target will have to move inside the polymer in order to hybridize. And same goes if you're working with antibody, then we must make sure that it does not cover the site where the epitopes will interact with the antigen. We must make sure that only the lower part is being attached to. Okay, so that will be the one that is very important for you to think about, to characterize, to check whether the polymer is not covering the whole receptors and they are just good enough, just nice. How do we know that? If we have to test, look at the signal. Look at the signal. We get reproducible, repeatable signal, then it's okay. Okay, so using polymer is quite good because polymer is something that you can just buy and then dissolve and then put on top of the sensor surface. Okay, but the possibility of the target DNA not be able to diffuse into the polymer is there. So we have to think about whether it's good for our sensor or it's not. Yeah. So this is how the polymer interaction, as I mentioned just now, phosphate backbone is attracted to positively charged surface, and these polymers, they are positive in charge. Yeah. So it can be like this, or it can be, yeah, it can be the bioreceptors lock in the polymer porosity, or we can have it interacted with the surface, the functional groups, the charges, yeah, of the polymers. Okay. So we have to think whether this will happen or will not happen. Okay, it's quite difficult. I have done this and the signal is not that good. Okay. For immobilization through polymer membrane to work effectively requires the probe DNA to be entrapped within the polymer such that it can't diffuse. Okay, of course. But uh, preventing the DNA being able to diffuse also precludes target DNA, okay, from being able to diffuse. Hence, only DNA the surface is involved in hybridization. So there is a big possibility it's not suitable for DNA. Now, I think we have the biotin first. Let's go to the oh silicon surface. Right. Okay. This one that I mentioned, which I do not have the slide, I miss it probably, is the biotin streptavidin chemistry. So if I miss this, I will show you next week. Okay. So now let's go to the covalent attachment. Okay. So let remind me to show you on the biotin streptavidin strategy, which is quite common. All right. So covalent attachment is about covalent bond. Okay. It depends on what is the linker. Okay, remember about DNA probe that I have shown you this table. What is the linker that exists? Okay, in the probe. Okay, the linker, okay, can be it's up to you. It can be customized. Yeah, whether you want this is thiol linker. This is what we call as aminated DNA. Okay, because it's amine link. This is also amine. Okay, it can be customized. Because we usually order from companies. Yeah, it's a chemical synthesis on how to attach thiol to the DNA probe. Okay, so even the production of DNA probe is chemistry. It's just chemistry. Yeah, it's like attaching a series of amino acids because you know already the sequence, and then you put in the linker as well. So this is very important to identify before you can choose the covalent method. Okay, if you have just this long chain of amino acid, you cannot do the attachment by using covalent. Yeah, you need to have this linker. Okay, right. So let's look at if we have aminated DNA. So the DNA probe has this amine group at the end of the uh, long chain. Okay, let's say, and we are using carbon. Okay, we are using carbon because different material will have different strategy. Okay, let's say we have aminated DNA and then we have carbon. So just now, if you look at here, we have, let's say we use this and we have carbon. So this carbon will not be able to uh, um, naturally react with amine. Okay, it cannot be naturally react with amine. So we have to do some chemical modification first. Okay, so you can see that uh, we have this, what we call as amide chemistry. Amide chemistry. So what is amide chemistry? Is we are going to attach carboxyl with nitrogen from the amine. Okay, so you can see here. Okay, so we are dealing with amide chemistry. So if you look at from the strategy, you can see that there is extra chemical reagent here. Why? Because we need to add carboxyl group first. We need to add carboxyl group first on top of the carbon. Okay, because carbon is just carbon, we need to activate the carbon, okay, with a carboxyl group. We can do acid, strong acid, so that the carbon will be activated with carboxyl group. But in this case, we have this carboxylic acid, and once we have activated the carbon, we can see that this carbon surface now have carboxyl group at the end of on the surface of the carbon. So what will happen now is we need to attach this carboxyl functional group with the amine of the linker amine on the DNA. Okay, so we need to have what we call as EDC NHS activation. Okay, naturally, this carboxyl will react with the amine, forming amide chemistry, this one. Naturally, it will. But it will be reversible. So we do not want reversible. We want to be a stable amide chemistry. Okay. Just now. Okay. So the EDCNHS, okay, is very common. Okay, you can just Google EDCNHS chemistry. Okay. EDCNHS activation. This is very common thing. When we do amide chemistry, we use EDCNHS activation, okay, to get the irreversible amide linkage. Okay. So now once you see, we cannot see. Once we have the amide, stable amide chemistry, now we have the DNA strongly attached to the carbon. Okay? So every materials requires different chemistry. Okay, so this is carbon and we have aminated DNA. So let's look at another example. So this is again carbon, okay? And this is an example if you are using nanoparticle, okay? And let's say you have carbon nanotubes, okay? Nanomaterial, for example. Nanomaterial is easy to group them in nanomaterial. So we have carbon nanotubes, let's say. So as we know, carbon is just carbon before it is activated. So the first step is to activate with acid. Okay, activate with acid. And then, of course, like just now, we need to, with activation of acid, we can have the carboxylic group, carboxylic group on the surface of the carbon. Same goes with the activation is actually EDCNHS, the EDAC. Okay. Now we are able to link the whole thing with NH. Yeah. So this is the final how it looks. So if you have any proteins, not necessarily uh, DNA, because protein does have amine. So this is among the best strategy to attach the proteins. Yeah. And chance EDC. Yeah. Okay. So let's say we have gold. Instead of carbon, we have gold. Okay. So what chemistry can we use for gold? So one thing about gold, okay, they have this very special binding ability with thiol. They have very special binding affinity with thiol. It is not covalent, but it is considered pseudo-covalent. You can read about it, pseudo-covalent. So it's not really covalent, but it's a pseudo-covalent. So let's say what do we have here? This is very easy. Why? You can see that with, let's say, let's look at example with thiolated DNA. Yeah, thiolated DNA probe, where you have the thiol group at the end of the DNA probe. So what you can just do is just incubate. Yeah, incubate the gold with the thiolated DNA, and then it will naturally be uh, attached to the surface of the gold. Yeah, because of the special binding affinity between thiol and gold. Yeah, it's just an incubation. Um, yeah, we can use automated machines, but we can do simple incubation. Just incubate the gold surface together with the thiolated DNA. Now, this one, if you look at here, we have like longer version. Yeah. Okay, I will explain that why, because sometimes we need to extend a bit. Why we need not just a simple one, but we need longer. You can see that is getting longer than the the normal one. There is uh, um advantage and disadvantage on this kind of. I will explain why we need longer. So the last one is actually the same principle. When we say about SAM, is actually thiolated DNA, okay, thiolated DNA with self-assembled monolayer. Sorry, with gold surface. Yeah, with gold surface. So now this is why you can see that there is uh, additional strategies of chemistry involved. Yeah? So why is it we cannot just stop at incubation? Okay. Simple incubation because of this issue. Okay. Again, it is related to orientation. Yeah, orientation of the bioreceptors. Okay. We want, okay, the red dots represent the thiol. Yeah? So by right, we want this. This is the correct orientation. The thiol group will attach to the surface of the gold, and the DNA is sort of standing up. Okay. But since DNA has negative charges, they sometimes behave like this. So we do not want this to happen. We do not want this to happen because it will affect the signal that we get from the sensor. So what can we do to avoid this issue is by using substrate passivation. What does it mean? We have to modify the surface of the gold with other chemicals. So this is what we call as alkene thiol. Okay, so this is alkene. And then this is thiol. So it is, you can use different kind of alkene thiol compound. And how does it work is because of the charges of alkene thiol is negative. Okay, it sort of make sure that the DNA to stand up. Okay, so it passivates the surface of the gold to prevent the DNA to lie down or to have different kind of orientation. So it helps because of the charges repel, because alkene thiol is negative in charge and DNA also negative in charge. So they have repel, so they stand up. Okay, so this is called substrate passivation. So how do we do that? Again, it's an incubation process. First, we incubate with alkene thiol. Okay, and then the next one we incubate with, so this is the gold surface. Later we incubate with the thiolated DNA. So this process requires optimization. How long we need to incubate, how much alkene thiol we can add, how much thiolated DNA we need to add. So this one has to be studied. We do not know how much actually, but we based on the signal generation. Okay, so remember that if it is too much on top of the surface, it will restrict. Yeah, it has some issues with restriction. Okay, so it cannot be too much. It cannot be too low. So it has to be studied. Yeah? So this is the full protocol on how to do self-assembled monolayer, right? So you can see there's a lot of incubation involved and cleaning of electrode. In this case, we are talking about electrode. Okay, this is the protocol using aminated DNA. Okay, if you're interested, you can read in detail of how we do that. So the last surface will be silicon. Okay, carbon, gold, silicon, because these are the common that we use in sensor development. Okay, so let me show the cartoon of how we can do chemical modification of silicon in order to attach bioreceptors. Yeah. So the first one, okay. So this is the silicon surface, right? And in order to get the first, usually we treat it with sodium hydroxide. Why? Because we need to have, right? Okay, this group first, the hydroxyl group. Okay, because silicon is just silicon unless treated with alkaline, strong alkaline solution, then we can have the hydroxyl group. Yeah. So after that, we use epters. I forgot the full name of epters, but the end is tetraorthosilicates. Yeah. So with that, you can have the amine group. Okay. So with the epters, you can get amine group on top of the silicon surface just now that has the hydroxyl group. Yeah. Now, if you can just stop there, because amine, let's say you want to attach with something that is suitable with amine, it's okay. But for DNA, because in this case, let's say we are using aminated DNA. Okay, remember about aminated DNA, the DNA probe has the amine linker at the end. So amine cannot react with amine. So we cannot stop here. We need to use, okay, this is the first step just now. With apters, amino, I don't remember the middle, but I remember the tetraorthosilicate. You get the amine group. And then next one, why just not coming? Okay. So the next one is in order to have the amide chemistry just now. Yeah, let me, okay. Yeah. Okay. So we need to have glutaraldehyde. Yeah. So glutaraldehyde will have, we can have the amide chemistry. So you can see this is the N from NH2, and then it will interact with the carboxyl ketone from the aldehyde group. Sorry, this is aldehyde group. Sorry, aldehyde group. So this is what we get from this amine and aldehyde group reaction. Okay, so the final objective is to attach the DNA biomolecule with aminated DNA is possible because we already have the aldehyde group. Okay, so it's not just carboxyl. We can also use aldehyde group to react with the amine linker of the DNA pool. So you can see there are a lot of chemistry involved, and it depends on what you have, what material, what linker. We have to design the chemistry step by step, right? Any questions? So this is again the same thing, but now is in chemistry form. Sorry, this is a bit more on same process. We have silicates, and then we have with thiol. Yeah, there's a lot of chemistry involved. Okay. I think I put it here. Okay. Right. Okay, another 5 minutes. Um, so these are the steps that you can use if you have silicon surface. So up test, okay. Incubation with up test, and then the rinsing, the washing, and then you put glutaraldehyde, and then the rinsing, and finally incubate with the aminated probe. So this is, I have introduced to you the process. Yeah. So carboxyl aldehyde group can also be used to react with the amine. So another one big issue is the last part we mentioned about non-specific binding. There is a possibility of other DNA apart from the target, other protein, or any other interference that can inhibit the actual sort of, it is unwanted interaction. We do not want the interaction to happen, but it happened. Okay. So how do we solve this? Okay. One thing, remember about the SAM just now, we can use alkane thiol, okay, or mercaptohexanol, as long as, yeah, it will have a negative charge on top of the sensor surface, and it will make sure that the DNA to stand up. So this is also considered a strategy to avoid non-specific adsorption. Because why? Because if there is other protein, unwanted protein to come here, it will be repelled. But it will also repel the protein that we want. So this is actually quite difficult, but we can do washing, okay? Because remember about if DNA is 100% hybridized, it is a strong one. It is a strong interaction. But if you have like 10% hybridized, comes from non-target, different species. If we do washing, then it might be able to detach. Okay, it might be able to detach, sort of clean the unwanted interaction. Yeah. Okay. So the other one is we can use longer linkers. Yeah, you can see the linkers here. They are different length. The so this also affect the signal. Okay, so we can study the length of linkers and how long is this is called diluent to avoid the DNA from from lying down. And this is the linkers. So this also affect the signal generation, how good the signal, how reproduce, how repeatable, okay? So another one is about the temperature effect, okay? pH also very important. Okay, so usually temperature we study from 37 to 40 degrees. pH usually we know it is going to be around 7 to 8 because this is the pH of biomolecules, right? Density, okay, we have talked about this. Yeah. Right, so okay. So now the last part, as I don't know, some of you were not here. I talk about from 8 to 9. Okay, there'll be no formal class, but you have to access the Putra Blast. I have put the lecture slides and then recorded videos on electrochemistry. Okay, why? Because I need you to read first, because electrochemistry is a bit difficult. Okay, for a new a new people. I'm not sure you have done electrochemistry before during your bachelor's or during your, I don't know, during your undergraduate study, especially I don't, some of you are doing PhD, so probably you have done master, you knew something about electrochemistry, but I need you to read first before our discussion on electrochemistry next week. Please do that. Okay? All right. So in these few minutes, I just want to test on the how we can group you into different room. Okay. Why? Because I need you to get to know your member before we can do group practice next week. Okay, so I'm doing breakout rooms. Okay, so you will be randomly put into the breakout rooms. So there are 11 of you, no, -2, so 9 of you. So I'm going to do 3 big breakout rooms, and you'll be in the breakout rooms for a while. Just get to know who's your member, and then we regroup back for further instruction. Okay? Okay, all right. So 3 breakout rooms. Right. So you need to move Okay, now I can see that in room 1 we have SD. I forgot your name. Helmi and Valerie, right? Okay. Room 2 we have Hazira, Uche, and Hanya. Hanya. And room 3 we have Atika, Farah, and Tiffany. Right? So get to know. Just know yet who's going to be in your groups. Okay. Yeah. So this one will be the next lecture. We are going to do some exercises in group and you'll be presenting. Yeah. So now I close all the rooms. Everybody will come back to the main session.